Hello and welcome to the latest Technology Networks webinar, Building Better AI with High-Resolution Functional Genomics Data. I'm your moderator, Dr. Stephen Gibney, Science Writer for Technology Networks, and I'm excited to be here to host today's sessions. We have a fantastic presenter, Dr. Chu, who will be sharing some of his valuable insights with us. Dr. Chu is the Vice President of Early Discovery at Zyra Therapeutics, bringing a decade of experience at the intersection of high-throughput biology and AI, MLM-driven discovery biology. His research has leveraged complex human iPSC disease models in neurological and metabolic indications, combining pooled transcriptomic and optical screening with state-of-the-art machine learning to illuminate novel biology. Dr. Chu is an accomplished scientist with 5 patents and many publications across multiple journals, including Cell and Nature. After his presentation, he's kind enough to offer us some time for a Q&A session, and we encourage you to submit your questions at any point during the presentation. To do so, just type your questions into the box on the right-hand side of the screen and click Send. We'll do our best to address as many questions as possible in the time that we've got available today. If you encounter any technical difficulties, please click the chat box on the right-hand side and click Send to request assistance from our support team. Without further introduction, I'll now hand over to our speaker, Dr. Chu. Over to you.
Great. Thank you, Steve, for the introduction. Can you hear me okay online?
We're all good here.
That's great. Well, good morning, good afternoon, good evening, wherever you are. And my name is Chu. I go by Chu, my last name, because my first name is very hard to say. So just call me Chu, as in Chewbacca. I'm the VP of Early Discovery at Zyra, and I lead the High Throughput Biology group, supporting both our protein design AI efforts, as well as our virtual cell AI efforts. Today, my talk will be mainly focused on virtual cell, our vision, and our recognition that the field is data poor, and we need to generate a lot more data, and how we are going to go about doing this. So happy to share with you our thinking and our latest work on that front, and looking forward to chatting with you afterwards about it. And let me just progress the slides here. Okay. So at Zyra, we're focused on building a foundation model of biology, or bio-AI. And we think a causal model of biology will be incredibly useful for drug discovery. How we think about going about this is starting with biological models at different scale or layer of biology. Cells in the tissue culture, animal models, all the way to human samples. Perform high-throughput experimentation or perturbation on these models, generate biological data at massive scale, and then feeding this data into training a model that understands cause and effect, perturbation and outcome. We think such a causally aware model will be very useful for predicting perturbational outcomes of the cell. How do we turn cell from state A to state B, from healthy to sick, sick to healthy, which obviously will be very useful for mapping or evaluating targets, as well as elucidating target epitopes for drug intervention. But we also think such a foundation model would have multiple uses. For example, mapping mechanism of actions, which would allow us to match drugs to patients of the right phenotype, as well as matching different disease models to the right indication for research. And last but not the least, such a model will be, such a foundation model should be able to generalize different contexts. And this last point is very crucial, because obviously the ultimate goal of drug discovery is to predict human response to drug interventions. But human response, or even animal studies, are incredibly difficult to scale in the lab. On the other extreme, simpler models, such as cells grown in the tissue culture, is very easy to scale for massive data generation, but these data tend to be less translationally relevant. And this gap, this translational gap between simpler models and human response prediction is a big chasm. Today, the gap is often bridged by human intuition, expert reasoning, and frankly, a whole lot of trial and error that are expensive in the lab and a clinic. And if we can build a model that can be pre-trained on large amounts of data generated from simpler models, fine-tuned on scarcer data from increasingly more complex but more translationally relevant models such as primary cells, organoids, animals, and eventually from human. This model hopefully will be able to transfer learn the context and give you predictions in more translationally useful systems, including eventually be able to predict human level response. I think that's really the holy grail in this field. Excuse me. So that's obviously a very ambitious goal, but we're encouraged and we think the time is now by innovations that's happening in the consumer AI space. So there, as everyone's aware of, the explosion of data, all of internet's text, as well as algorithmic advancements, for example, transformer architecture, has really ushered in a revolutionary in this space. And through a relatively simple pre-training task, for example, predicting the next word or next token in a sentence, the model can efficiently learn human grammar, syntax, and knowledge. And then through relatively fewer data for fine-tuning, for example, with high-quality question and answer pairs, or with human reinforcement learning feedback, the model will be shaped into a very helpful chatbot. And that's, of course, the ChatGPT and the Gemini that all of us use today. We actually see a striking parallel in the biology space as well. Models have been trained on large amounts of single-cell RNA-seq data, for example, as demonstrated by pioneering work in this field, SCGPT, which is invented in Dr. Bo Wan's lab from University of Toronto, who's now recently joined as our head of biomedical AI. These transformer architecture models can be similarly trained on large amounts of single-cell RNA-seq data. And they have pretty impressive performance on a series of descriptive tasks such as cell type annotation, batch effect normalization, and so on and so forth. But one important caveat is that these models don't yet outperform simpler models on perturbational tasks, predicting what would happen to the cell if I intervene in a certain way. And you can see the problem. These perturbational tasks are probably the more useful tasks to do in drug development. And that's an interesting thought problem to have. Why are these models that's trained on such massive data set not able to outperform simpler models in these perturbation prediction problems? And we think the problem starts with the data. The data that are used in these model training are often curated descriptive data sets, for example, cell by gene from CZI that are curated from many healthy human donor sample studies. There's a lot of redundancy in these data, a lot of PBMCs, a lot of brain samples, and there's not very much disease context yet, and very little perturbational context. And so, of course, a model doesn't know what it doesn't know if it's only seeing profiling or descriptive data. And we think a big gap in this data that's missing is perturbational data, where you intervene a cell in a certain way and measure the outcome. We frankly think that that's a big opportunity for us to fill in that gap. So how do we think about data generation? Well, there are many techniques to do that. And we think one of the most mature way is perturb-seq, which combines pooled CRISPR screening with single-cell RNA-seq. Genome scale perturb-seq has been published in academia, but it's not straightforward to scale that to routinely to do large-scale experiments. And scientists at Zera really putting a lot of effort trying to scale and industrialize this platform by solving many challenges, all the way from cell fixation to fluorescent cell sorting, cryopreservation, figure out a way to do superloading of the microfluidic chips, all the way to end-to-end automation of the library preparation. I'll walk you through these improvements in the next few slides. But the end result is a high quality, what I call 2D data sets. On one dimension, you have high throughput perturbation because of the CRISPR screening done in a pool format. On the other dimension, you have high dimensional measurements because you do single-cell RNA-seq. And I think it's really these rich 2D data sets that are massive and high quality that can deliver on the foundation model of biology, just similar to how massive, rich, and high-quality 2D datasets like PDB, right, protein structures, have delivered for the field of protein structure prediction. Okay, well, many of these, let's start with fixation optimization. Many of these perturb-seq data are often generated using fresh cells, not fixed cells. And I think that's a big limiting factor. Think about the mechanics of doing these experiments. When you scale to genome scale, we're talking about harvesting tens, if not hundreds of millions of cells in a lab. And if you work on fresh cells, within a single day, you have to process these cells, put them through fact sorting selection, and commit to single-cell library preparation all within the same day. And that can be a very long day, often stretching over 12 hours. You can imagine the quality of the cell and after that long processing step. Many of them probably have incurred stress signals, accumulation, or even have died or on the way of dying. And that's going to confound your machine learning analysis downstream. And so we realized in the beginning that we have to figure out a way to fix the cell and not use fresh cells for these large-scale profiling experiments. But how do we do that? We actually tried many common fixative used in the genomics field: formaldehyde, methanol, and DSP. We found formaldehyde and methanol to be not quite compatible with either the single-cell library prep platform that we use, which is 10x GEMX here, or compatible with fluorescent-based cell sorting. DSP, however, is a reversible cross-linker thanks to the disulfide bridge in the middle of the chemical structure, and is readily reversed upon the reverse transcription, which happens during a reducing condition. So we found that to produce really high-quality RNA-seq. And whether we fix the cells before sorting or sort the cells before fixation, we found the data sensitivity and the gene expression level to be highly correlated, which is really convenient. And in practice, we found these fixation by DSP produce comparable, if not better, data quality in our own experiments shown in blue and the pink than the industry average data set, which is a reproducible data set in K562 that was a first genome scale perturbation down from the Jonathan Weissman's lab. But perhaps equally important is that this fixation step allows us to not have to commit to a genome scale perturb-seq on the day of the cell harvest, which is really important because you can imagine spending hundreds of thousands of dollars in reagents, committing to the single-cell library prep before you know if the experiments even is high quality or not. And being able to fix and time shift the assay allows us to take a small aliquot of the cell harvested, go through with the whole library prep and sequencing, make sure that the data quality is good before we really commit all the reagents and cells for the scaling up to the genome scale preparation, which is very handy. Along the way, we also figure out other tricks. For example, that we can superload the 10x microfluidic chips up to 5x, reaching about 100,000 cell scale per channel, which really help with cost saving, but also streamline the processing because now we have to do 5x fewer batches of experiments. And last but not least, when you're doing experiments at genome scale, we're talking about preparing hundreds of libraries per experiment, and that's a lot of work. And so we have automated the library prep part of the workflow with Hamilton Vantage equipment so that we make the work process much more streamlined, but also removing the possibility of human errors and variability in the data generation. So how does this look in actual practice? Well, here are our data shown from two genome-wide perturb-seq, one done in HCT116, which is a colorectal cancer line often used in oncology research. The other is in HEC293 T cells, often used in drug discovery. And so it actually worked much better than even our wildest imagination. So this fixation plus cryo storage condition in the presence of RNase inhibitor actually made the RNA stable all the way up to 140 days in cryo storage, which is really useful. And that means that we can perform these experiments over multiple batches. We don't have to cram all of the library prep and cell thawing and sorting all into a single day. So here we're showing that both the HCT116 as well as the 293T genome perturbs are done in many different batches over many days, spread over that period of time, which really help the team prepare and plan for their experiments and not have to rush things in the lab. And the end result is much higher data quality. So here I'm showing you the UMI detected per batch per cell, which reflects the unique transcripts that we can detect in these cells. And in single-cell RNA-seq, data are notoriously sparse sometimes. The more unique transcript you can detect per cell, the higher sensitivity you have in the assay, the better chance you have detecting more subtle and nuanced biology. You can see that the Zara data shown in pink and blue outperforms that of the industry average reproducible data set, and is almost 10 times the sensitivity of another large-scale perturbation data set, Tahoe 100 million, published recently. And not only that, Zara data also shows superior inter-batch variability. And so here we're measuring the correlation between experiments done in different batches. You can see that the Zara data is much more tightly correlated than the reproducible data previously published. And the question we often get in data generation is, in single-cell RNA-seq data, how broadly universal is it to capture perturbational effects? And we're very happy to see that both in our own data as well as in reproducible data, the answer seems to be RNA-seq is broadly applicable. So we can detect anywhere between 40 to 50% of our perturbations done in expressed genes in these cells. We can, by training a linear classifier, we can detect statistically significant perturbational effects in these cells, suggesting that single cell is a pretty useful, broadly applicable assay to capture perturbational effects in these cells. Not only that, the single-cell RNA-seq measurements are also semantically meaningful. So here we show that perturbations that have higher correlations measured in perturb-seq data also tend to have higher evidence for protein-protein interactions as curated in this database for PPI, which is the string DB, showing that known protein-protein interactions or known biology complexes are indeed captured by our perturbational correlation measurements. To illustrate this in a graphical way, we can take the top perturbations in our data, represented by the columns in this heat map, and cluster them by the top variable gene expression in our data, which is shown by the rows in this heat map. You can see that a simple clustering readily reveal known protein complexes and cluster proteins of similar functions together. You can see that exosome complex, genes involved in ribosome biogenesis, protein synthesis, and nuclear pore and mediator complex all jump out immediately from this simple clustering analysis. Perhaps the most striking and interesting finding from our data when we have these genome scale measurements done in different cell lines is this observation in the CRISPR knockdown that we use in these measurements. And if we can find a way to leverage that information, then you can begin to potentially understand dose-dependent gene effects in the cell. And that will be incredibly powerful because everything in drug research is, of course, all about the dose. But direct measurements of the target gene knockdown is very difficult because, as I mentioned earlier, single-cell RNA measurements can be pretty sparse. So on a per-cell level, it's not reliable to look at just the primary transcript expression level to infer its level of knockdown. And would there be a useful proxy measurements that we can use to stratify cells with different knockdowns? We looked deep into our data sets and found that the guide RNA expression, which is reliably captured, a polymerase III transcript, reliably captured at hundreds of molecules per cell often, actually correlates pretty well with knockdown levels. The higher the guide RNA expression you have in a cell, the higher the knockdown tend to be in a cell, which, if you think about it, makes a lot of sense. CRISPR inhibition, of course, is mediated by two enzymatic components, the Cas9 enzyme and the guide RNA in the cell. And it would make a lot of sense. The more guide RNA you have, the higher perturbation effect that you have. And we show that you can readily stratify cells with high guide RNA and low guide RNA, and they actually correspond to higher level of on-target knockdown and lower number of on-target knockdown. And this is quite helpful because you can use it to stratify and filter away cells with poor knockdown, and there's natural heterogeneity in any large-scale CRISPR experiments, and only keep the high-quality cells for downstream analyses, or you can use them to perform dose-dependent analyses. And we show that that's possible as well. The low knockdown cells stratified by low guide RNAs, shown in light blue, have lower perturbational effects as measured by energy distance than the high knockdown cells as stratified by the high guide RNA, shown in the dark blue. So we envision that this could be a very powerful proxy measurements to help us filter away cells with poor knockdown or stratify cells into different knockdown and secondary effect categories, which potentially could be helpful for training a foundation model of biology that has dose-aware information. We share the data sets and as well as the fixed perturb-seq method with the community back in June in a preprint, recognizing that the whole field is data poor, and having some high-quality data in additional cell lines than KPX2 will hopefully jumpstart the whole field of building foundation model biology. We also recognize that it's not just the scale of the data, quality matters a lot too, and hopefully this preprint helped raise the bar for quality in this field, and the fixed perturb-seq method would allow others in the field to generate similarly high-quality data sets. Since then, we're quite heartened by the public reception. Our data set in just 3 months has been downloaded more than 64,000 times. And we've been received attention from multiple industry outlets, 5 featured stories in industry news, as well as some very nice call-outs from industry key opinion leaders, Dr. Emma Lundberg, for example, who's the co-director of the Human Protein Atlas. This data is licensed under non-commercial license, so it's freely for use by any academic labs or consortium, and we're happy to chat about commercial data licensing for our data sets. Of course, internally, we're not stopping there. The 2 cell line data is only the tip of the iceberg. That was done in Q1 2025. Internally, we are expanding data generation already to many other cellular contexts and going beyond cell lines into primary cells, iPSC and iPSC-derived cell types, as well as performing direct in vivo pooled perturb-seq. And we think the diversity, the quality, and the scale for data will truly complement the descriptive data that's out there and together help usher in the next generation of foundation model of biology. But I just want to say that lab data generation is only half of the equation. We think combining lab data as well as patient models built on patient data that others in Zera are working on will together integrate into insights that will help deliver the next generation of precision medicine. I won't have the time to talk about it today, but hopefully we'll have more to share on that soon. So in summary, today I share with you hopefully our vision that perturbation data is critical for building causal foundation models of biology. And at Zera, we have built an industrialized platform to generate high quality and massive scale of these perturbational data using fixed perturb-seq platform. And we think that broad perturbational effects can be captured by perturb-seq, and that these data can be leveraged for dose-dependent analyses. This work is only possible with a highly talented and dedicated cross-disciplinary team of people at Zera. So spanning across functional genomics teams, our amazing genomics core, our bioinformatics and automation teams. And today I was only able to talk about the virtual cell effort, but of course at Zera, we are an end-to-end AI drug discovery company working on many other AI efforts, including protein design, as well as drug development and patient models. We are rapidly expanding, so please check out our website if you're interested to look for relevant positions, and we're happy to share more on other efforts in the future. So I'll stop there and happy to take any questions.
High throughput screening is fundamental to biomedical research, particularly for the development of protein-based therapeutics. By iteratively screening protein variants, researchers explore a protein's vast sequence space in search of a novel variant with desired, optimized properties. However, finding the right protein variant is difficult, even with the latest AI tools. Large-scale screens and training datasets for machine learning require pools of DNA, encoding thousands of defined protein variants to effectively sample the protein sequence space. There are many ways that researchers can generate their screening libraries, but current technologies leave much of the sequence space out of reach and underexplored. If your protein is longer than 300 nucleotides, your options for building screens are either very expensive, very limited, or very biased. As a result, the sequence space of most known protein sequences cannot be effectively studied in high throughput. To advance therapeutic protein development and AI-driven screening, researchers need tools to economically synthesize longer sequences at scale. At Twist, we constantly ask, how can we give more scientists access to longer synthetic DNA on a large scale? Our team used our proprietary silicon-based platform, optimized chemistry and automation to become the only company able to synthesize precisely designed pooled sequences that push the boundary beyond 300 nucleotides. This innovative new product, called multiplex gene fragments, consists of uniform pools of double-stranded DNA that are designed to your sequence specifications. In just 1 week, you can receive pooled lyophilized gene fragments, up to 500 base pairs ready for use in your high-throughput screen. With access to significantly more sequence space, you could precisely synthesize a library to generate training datasets to improve your machine learning model, or screen single-domain heavy-chain antibody variable regions and make discoveries that will lead to the next blockbuster protein therapeutic. So, what will you do with significantly more sequence space? Get started with multiplex gene fragments today.
Thank you everyone for your attention, and thank you Chu for your incredible presentation. We're now going to move on to the Q&A session. As mentioned at the start, please feel free to submit your questions using the box on the right-hand side of the screen. Simply click send, and we'll do our best to address as many questions as we can in the rest of today's session. We've already had a few questions, so it's okay with you, Chu. I think we're going to dive straight in.
Of course.
Yeah, fantastic. So the first we've had is, what are the limitations or trade-offs of the fixed platform, if any, especially when scaling to this level?
Yeah, that's a good question. We have not found many downsides to the fixed platform compared to using fresh cells. And I think when you're anyone who's performed large-scale perturb-seq will readily realize that it's really difficult to do so with fresh cells. And the fixed platform really buys you the peace of mind and the time, the poise to plan these and streamline the operation. And we show that the beauty of the DSP fixative reagents is that it can be readily reversed during the RT step. And so it's truly compatible. We haven't seen many loss in sensitivity of RNA-seq. Maybe the only word of advice I'll give to people who are performing these large-scale experiments using DSP fixative is that it's a pretty gentle fix. maybe gentler than formaldehyde or other fixative agents. So when you're handling the cells, just try to be gentle and don't triturate too hard. These are not an extensive fix as a soft fix, I would say. And so just putting the same care that you would in handling live cells, and you will do okay.
Fantastic. That makes a lot of sense. Again, good practical tip there of being delicate. That makes sense. We've had two questions that are a little bit similar here. So the first is, can you speak to the types of perturbations supported, CRISPR-I, CRISPR-A, or any others, and how that impacts the resolution of the data?
Yeah, this platform of using a fixed approach to perform large-scale perturbation is really agnostic to the CRISPR modality. Here, we only showed CRISPR-I knockdowns, but this would similarly apply to CRISPR nuclease knockouts, CRISPR-A, or other modalities. I didn't have time to go into the details here, but the way we deliver the CRISPR guide RNAs are using a tRNA spacer for dual guide delivery. And we found internally that using two different guide RNAs against the same target promoter allows you to achieve a bit better knockdown than using just a single guide. And also, you can imagine this be a pretty seamless platform for extending from single plex perturbations to dual plex perturbations, simply by directing the guides not against the same promoter, but two different promoters for two different genes. So we think the platform will be quite amenable to performing combinatorial CRISPR perturbations in the near future.
Fantastic. And kind of similar lines here, another question in terms of other options. So someone said, are there any plans to correct other omics data, such as SC, ATAC?
Yeah, that's a great question. So we started with perturb-seq, mainly perturb single-cell RNA-seq, because that is the most mature way to scale data generation in this field. There are many other ways to generate these kind of 2D data sets that are high throughput perturbation on one axis and high dimensional measurements on the other axis. Examples include exactly like the person asked. Single-cell ataxic data will be quite interesting. Other data sets include high dimensional, high content imaging data. In my previous life, I worked on pooled optical screening platforms, which combines, again, single-cell high throughput pooled CRISPR perturbation with cell painting assays or other live cell imaging methods. So we think all of these data modalities could be quite interesting. Ultimately, I think we will have to look at the benchmark model performance to know which data give us the most unique or diverse bits of information per dollar spent.
Yeah, that makes a lot of sense. We've got a few minutes left, so maybe one more question, a bit of more of a bigger picture one here. What would you say the biggest challenges are in the generation of foundational models that still need to be overcome?
Yeah, that's a great question. As I alluded to earlier, something that we didn't go into today, but I really think is the key to advancing this field is actually setting up the correct metrics, the benchmarks for evaluating a foundational model biology. There are many open questions in this field. For example, exactly like the previous question, what type of data would be the most useful to achieve foundational model biology? How much data is needed? And how do we best generate high diversity data? I think these are questions that can only be answered if we first set up as a field quantitative and widely adopted benchmarks. And without these benchmarks, I cannot see how a team would be able to internally understand if they are making progress, or how do we evaluate work from different teams and learn from each other. And a good analogy maybe is what's happening in the consumer AI LLM space. There, I want to make the distinction between a technical loss used in training, for example, predicting the next token. That's very useful for training these LLMs. But as consumers of ChatGPT, we don't really care too much about how well the models predict the next token. For us, it's really how well can the models help us in our daily work, how well can they pass GRE exams or bar exams. And that's how we truly evaluate the model's ability to reason, to generalize, and to problem solve. And then perhaps a similar analogy can be made here. in training these foundation models, oftentimes people will use loss functions such as RNA-seq reconstruction loss. And that's great for training these models, but in daily use, biologists probably more care about metrics such as, can these models really help us identify novel combinatorial perturbations or generate ideas I haven't thought about before? So I think the sooner the field can come together and build that AP biology exam, so to speak, for these foundation models, the sooner we can start to measure quantitative progress in the field.
Those analogies seem like a great place to wrap up. And I think, as you said, I think everyone is really looking forward to seeing where this field is kind of going and what we can look forward to in the future. So that is all the time we have today. A big thank you to Chu for your time. Please remember that any questions that we didn't get to will be answered offline as soon as possible. You can also continue to ask questions even when watching this webinar on demand. Don't forget to download your certificate of attendance, available from the handouts tab on the right-hand side of the webinar platform. Once again, a big thank you to the audience for your participation, and an incredible thank you to Dr. Chu for your time today. It's been incredible talking to you. Thank you very much.
Thank you, everyone. It's been a pleasure.